sediment. And so when the peak is high, we have high amounts of those minerals, and when the peak is low, we have very little. And it is a very important way that allows us to take separate cores and match them to each other. So Scott pointed out there's one peak here in the Lungo 9 core for which they ran the pollen, and it allows us to match it up with later cores and even between the two lakes. And these are showing lines of correlation for smaller peaks between this 09 core, which is here, and then the new one that we took in 2012. So, as a geologist, I'm going to talk about two different types of data. One are diatoms, because I'm principally a micropaleontologist, so I work on fossil plankton, and diatoms is one of the principal groups that I work on. And then after that, I'll show you a little bit of the results in the basin on the sedimentology and the geochemistry. So let's start by talking about what diatoms are. They're a group of unicellular golden algae, and they're interesting to us in lake core studies because they produce a mineralized skeleton that's made out of opal silica. And this one is a live diatom, and you can barely see the siliceous skeleton here. And inside you can see the chloroplasts. And they also are very rich in lipids, in lipid droplets, and that helps them because the mineralized skeleton makes the diatom heavy, and it wants to sink to the bottom of the water, but then the lipids allow it to remain in suspension. And if we were to clean the sediment and look at the skeleton under the scanning electron microscope, we would see that it is a box shape made with two valves here that are then held together with a girdle band. So diatoms, modern diatoms, are found in virtually every aquatic environment. They're in the oceans, they're in lakes, rivers, springs, and they're very sensitive to a variety of changes. For example, pH, temperature, nutrients, all of these things determine which species are present. And as a result, we've been able to use them in both paleoecology and in climate studies. In terms of their ecology, I'm going to talk about two major habitats for diatoms. One of them is as attached algae, where they're living in shallow marshes and on the perimeter of the lake. And we call these periphyton, and they're very diverse. These golden strands here are colonies or chains of Starosira, and they will entangle themselves in the green algae, and they're generally found in water of depths of about 5 meters or less, and they can also live in rivers. And then there are other types of periphyton, such as the Gomphanema, that produce a stalk that's made out of a polysaccharide mucus, and that allows them to attach, and they can persist very well in streams and running water. So the diversity is very great, and a typical sample may have well over 100 different species. So part of our initial work is to just document the modern flora and then also try and identify them in the cores. And many of the identification criteria are based on the skeleton, so we can actually know what